our first speaker talks about how to use genetic engineering to make GFP as a imaging probe. Now I'm going to talk about just how to use traditional chemistry method to use to make imaging probe. So our probes are based on quantum dots, which are essentially a piece of semiconductor materials. So when you make semiconductor to be within nanometer size, we can get this so-called quantum confinement effect and a lot of wonderful new optical properties. For example, we can use just one single illumination source to get different colors at the same time. And the synthesis for these different colors of quantum dots is simply based on different size. We just change the reaction time of the synthesis chemistry, and we can get different size of quantum dots which corresponds to different colors. And the quantum dots are much brighter than dyes. The left vial contains a dye solution, and the middle and right vials contains different colors of quantum dots with exactly the same molar concentration as the dye solution. And you can see, I think this picture speaks for itself. And quantum dots are also much more stable against photobleaching. This is quantum dot. After 30 minutes, they basically don't photobleach at all, but with a conventional dye, it loses its fluorescence completely after just 2 minutes. However, I want to point out that quantum dots have limitations compared with fluorescent dyes and fluorescent proteins, mainly because quantum dots are much bigger than fluorescent proteins. So, for example, when you are trying to bind these quantum dots with your target, sometimes there is steric hindrance. So we are not trying to claim that quantum dots are going to replace fluorescent dyes or fluorescent proteins. Despite these limitations, people have been successfully using quantum dots for a wide spectrum of applications in vitro, in cells, in tissues, and in animals. So what our lab is trying to do is to develop a new generation of quantum dots. We are trying to get rid of blinking, and we are trying to make active quantum dot, which can serve not only as passive probe, but also perform some kind of effect on the molecule that it labels. And for this particular collaborative project, we are trying to make quantum dots to be switchable, which means that when we want it on, it can be on. We have considered two different strategies to make switchable quantum dots. Number one is based on doping. Doping literally means that you add some impurity material into the original semiconductor nanocrystals. And this impurity can bring in new energy levels, which can introduce this kind of switching effect. The other strategy is based on quenching. Basically, we add a secondary either nano object or molecular object, which can quench the fluorescence of quantum dot. And we apply some kind of mechanism, either by light or by binding, to change the distance between these two objects. This way we can switch the fluorescence on and off. So far we have got most of our results from this strategy. So this talk will focus on just the doping strategy. This is how we actually make doped quantum dots. It's based on crystallization process. Crystallization technique has been around for many, many years. However, what we do here differently from previous researchers is that we can make crystals of nano size. So the way we do this is to apply some kind of ligands that can coat on the crystal surface. This way we can slow down the crystallization process. For example, when you are doing general chemistry experiments, you encounter a lot of precipitation effect. So that's the traditional crystallization experiment, and that does not have a lot of control. So here we just apply some kind of ligands to slow down the crystallization process. For example, we can slow down the nucleation process by using a ligand called ODA. And here we apply manganese salt and inject this selenium solution into manganese salt solution. This way we form manganese selenium. Since we are applying this ligand here, we can slow down the nucleation step significantly. So even after 1 hour, we still just get the crystal nucleus. Then in the second step, we inject a zinc salt into the solution and slowly grow the crystal. And if we maintain this temperature at about 260 degrees Celsius, we can very precisely control the size of the crystal. And this is how the product looks like, just fresh out of the reaction. And you can see the fluorescence is in the yellow color range, and it's very bright. And this is how the spectrum looks like. The fluorescence is at about 585 nanometer. And the quantum yield is between 35 to 55%, depending on the batch number. This is the full width at half maximum, which is this value is about 50 nanometer. And the solution can be stable for at least 5 days. However, the quantum dots coming out of that reaction are not really soluble in water. In order to use them for biology, we need to make them soluble in water. So we have 2 different strategies to do this. Essentially, it's based on the same principle that you wash your hands. So if you have oil on your hands, if you directly use water, the oil tends to stay on your hand. But if you use some kind of detergent, it can lower the surface tension of the oil, and you can more easily get rid of the oil. Here we basically apply some kind of surface active agents. We either directly replace the original hydrophobic ligands on the quantum dot with a new ligand which has lower surface tension, or we apply a so-called micelle template to assemble several quantum dots into a new nanoparticle. Since the surface of the micelle is hydrophilic, this whole construct can be soluble in water. And also, because we are assembling several quantum dots into a new nanoparticle, this new nanoparticle is several times brighter than the original quantum dot. So this is the TEM image of the new nanoparticle. So as you can see, each micelle contains several dark spots. Each dark spot corresponds to one quantum dot. And these nanoparticles are pretty stable in water. After 15 days, the fluorescence is still pretty bright. So a key question is, of course, can we really modulate the fluorescence of these novel switchable nanoparticles, and the answer was yes. So when that should be off, actually. So when we don't have the modulation light on, the fluorescence of the particle is on. But when we turn on the modulation light, the fluorescence is off. And the decrease in fluorescence between these two can be as high as 80%. And the imaging was done by Peter's lab at University of Georgia. A surprising finding was that by using this micelle to assemble several different quantum dots into one new construct, it actually provides a protective layer against photo bleaching. So as I said before, compared with fluorescent dyes, quantum dots are much more stable. Now with this micelle protection, it actually gets even more stable. Here we apply a really high laser power to test the photo stability. As you can see, with traditional quantum dots, after 15 minutes, the fluorescence starts to go down. But with our micelle protection, the fluorescence stays on for at least 30 minutes. So in conclusion, we have got at least one strategy to make these switchable quantum dots working. And ongoing work includes, of course, firstly optimizing this doping strategy and try to integrate the quantum dots into two different biological systems by Guy and Beth. We are also trying to develop these quenching-based switchable quantum dots, and finally, we have been contributing to the QSTORM website. So I'd like to thank our collaborators and NSF for funding, and thanks for your attention.